Now that we discussed the important differences between eukaryotes, eubacteria, and archaeobacteria, let's return for a moment to discuss a common property of cells, the fact that they're usually very small, before we move on to discuss the methods that we can use to visualize and study cells. Cells come in different sizes. Some are very tiny, like mycoplasma, which is a type of bacteria that lack a cell wall and are about 0.2 microns in diameter, which is much smaller than a typical bacteria, which range from 1 to 2 micrometers. And a micrometer, for your reference, is a millionth of a meter. Other cells, like neurons, can be very large because they can have long projections that extend a meter or more away from the cell body. Nevertheless, most eukaryotic cells range between 10 to 100 microns, and usually they're around 20 to 30 micrometers in diameter. There are several reasons why cells are small. These include the need to maintain an adequate surface area to volume ratio for the exchange of materials, the amount of time that it takes for molecules to diffuse, and the need to maintain the right concentrations of reactants and enzymes that are needed for cells to function. In the majority of cases, the main limitation to cell size is the need to maintain an adequate surface area to volume ratio. The reason this is important is because the exchange of materials between a cell and its environment takes place at the cell surface. So as a cell increases in volume, the surface area needs to be enough to allow for efficient exchange of materials between the cell and its environment. The problem, however, is that as a cell increases in volume, the surface area of the cell does not increase at the same rate as its volume. So if you take a look at these cubes, these three have the same volume, the same total volume. However, if we divide a larger volume into smaller cubes, we have a greater surface area. So because the size of the cell will determine how much material the cell will need and how much waste it needs to get rid of, beyond a certain threshold, a large cell would not have a large enough surface area to allow for sufficient intake of nutrients and release of wastes. Cell size can increase as long as the membrane surface area is still adequate for the passage of materials into and out of the cell. One way of dealing with this problem is to change the morphology of the cell membrane to maximize the surface area. And that's typically done in cells that are specialized for absorption, like those that line the small intestine. Now, these cells, as you can see in this image, increase their surface area by having folds in the membrane. Now, these finger-like projections, which are known as microvilli, increase the effective membrane surface area because the cell now has additional surface compared to a cell that lacks these projections, and that in turn increases the ability of the cell to absorb nutrients. In addition to being limited by the need to maintain an adequate surface area to volume ratio, cell size is also limited by the rate at which molecules move in the cytoplasm. And the liquid component of the cytoplasm, which is known as the cytosol, is not like water. It's actually more viscous, like honey. So this further limits the movement of molecules. Now, many molecules move through the cytosol by diffusion. So that is, as you know, from an area of high concentration to an area of low concentration. And diffusion rates are affected by the size of a molecule. So larger molecules, macromolecules like proteins, are going to move more slowly. Now, in eukaryotes, this limitation to cell size is addressed by the use of transport mechanisms that require energy. Now, this could involve the use of carrier proteins, as well as the transport of material in membrane-enclosed vesicles that move along cytoskeletal fibers. Another limitation to cell size is the fact that cells need to maintain an appropriate concentration of reactants and enzymes, and the reason for this is that the rates of reactions depend on collisions between those reactants and those enzymes. Now, a way in which eukaryotes deal with this limitation is by compartmentalizing function. So what I mean by that is that the different organelles are specialized for different functions. So you only need to maintain a high concentration of certain enzymes and molecules within specific regions of the cell. So for example, the citric acid cycle takes place in the matrix of the mitochondria. So you only need to maintain high concentrations of the enzymes and reactants that are needed for this pathway within the mitochondrial matrix rather than in the entire cell. Now, although bacteria lack the organelles that we see in eukaryotes, recall that some bacteria possess internal networks of membranes. And bacteria also make use of macromolecular complexes that allow for functions to be localized to specific regions of the cell. However, the presence of many complex membrane-bound organelles in eukaryotes has allowed these cells to be generally larger than bacterial cells.